Thanks, Jamie. My name's Adrian McKemey. Despite the accent, I am actually resident in California, up in San Francisco. I moved here a few weeks ago so I could vote on the CERN ballot when it comes around. Apart from the R&D consulting side of things, I'm also responsible for the Stem Cell Center, which, in collaboration with CERN, we've been developing here in La Jolla to try and address unmet needs in the cell and gene therapeutic development area. So we're delighted to have a panel that moves the discussion to more of an international basis. And before I introduce the panel, I just want to give you an idea of the breadth of experience and content that will be resident here. We've got indications in neurodegenerative, cardiovascular, respiratory, macular degeneration, IBD, oncology, so broad range of indications. We've got functional representation for manufacturing, regulatory, strategic alliances, discovery. We've got companies that have been through every phase, from preclinical through to marketed products. We've got executives that have been through the whole company life cycle, from startup to acquisition. And we've also got, because of the international nature of this panel, representation from Europe, US, and Asia. So just a couple of ground rules before we get started. This was great because there was a lot of interaction amongst panelists. We want to keep that discourse going up. But also, in the sort of Quaker kind of idea, if the spirit moves you in the audience, stand up and yell out a question, because the more interactive this is, the better. We'd much rather have a deep discussion that got to an insight than to try and be comprehensive about the topics that we want to cover. I think that one of the things that we're hearing in the previous panel, and we may develop further in this panel, is the idea of some unique kinds of arbitrage that can happen in cell and gene therapies. We'll get to that as we go through the panel, but there may be some things that are different here, whether it's regulatory frameworks, manufacturing paradigms, other initiatives, which gives this particular field a kind of a way to not shortcut, but to accelerate certain parts of the normal drug development paradigm. We're going to focus on three kind of areas that cross-cut all of those functions and all of those therapeutic areas, all those geographies, regulatory, manufacturing, and innovation. But let me introduce the panel. First of all, Robert Deans from BlueRock. Come up, please. So Robert's Chief Technology Officer at BlueRock. Sometimes I get BlueRock and Third Rock confused because of the enormous, I think still record-breaking Series A that you guys attracted back a couple of years ago. And so Robert also has a particular interest in engineering out some of the immune response features in allo. So we'll get to that point of allo versus auto later in the discussion. Then Joseph Petrozello. So Joseph's from Brainstorm, where he's VP of Scientific and Corporate Communications, also previously and relatively previously at Juno. And so one of the things we're going to try and bridge is a little bit of an analog between what happens in CAR-Ts at Juno and what happens in neurodegenerative at Brainstorm. Then Hardy's chairman and CEO of Helios. So started off in ophthalmology, moving to other indications, but also has a particular perspective on the Sakigake approach, accelerated approach and regulatory framework in Japan, which we think might be interesting as a mechanism to accelerate ex-Japan assets moving more quickly back into their home markets. Gisèle. Thank you, Gisèle. Gisèle's Director of Project Management at MaSTherCell. And from this CMO perspective, although each of us may see our own particular problems and opportunities in our own companies, as a service provider, Gisèle sees them all in aggregate and has some perspectives about, at a systemic level, what some of the new approaches could be to accelerate development and manufacturing. Maria is from TiGenix. TiGenix has a very long history of work in this area. based in Belgium and now more recently in Spain, gone all the way through preclinical, all the way through to marketed product, and has very recently been acquired by, not Gisèle, not Maria alone, but the whole company, very recently been acquired by Takeda, so congratulations on that. And finally, Blake. Blake's from Cellular Dynamics, recently acquired by Fujifilm, and has a particular interest for iPSCs and about how they can be used for more predictive toxicology in stem cell research. So we're going to start, but like I say, if any of you feel an urgent question, just pop your hand up. We'll try and make sure that you'll notice and get to ask it. But first of all, Maria, from the perspective of having done all of this work in Europe, how different is it? How easy is it to get into the clinic with cell and gene therapies in Europe these days?
So thanks for the question. I mean, we heard already a lot in the previous session on issues in approvals, IRVs, and clinical protocols and everything. I think in Europe you have to add on that you have to go to X member states, so it's not only one shop with the FDA with your IND, but you will need to go to 15, 20 member states, and we see that that means complexity and delays in approval. So still, it's very amazing that we see very different feedback when we go to the different member states. I mean, it's true that they learn and they get alignment through the CAT, the Committee of Advanced Therapies at the European Medicines Agency, but still we see different feedback, so that gets complicated. Also in the introduction, we heard about GMO, which is a specific topic in Europe, so that also adds to the complexity. And then is the timing. It's also, specific schedules for ATMP. So instead of your 60-day, you'll get 90-day or extra, 180 days. So it takes very long. And then you have to sum it in these, site activations and the other things that we see, for example, in the US and other territories. So it's complex. It's true that we are positive in the new clinical trial regulation coming into Europe, so we hope that this is solving a bit the things. Also in the GMO front, there was this key meeting last week with the European Commission and the CAT. Things start to be more aligned as well, but still more to do. And definitely the BHP, the voluntary harmonization process, for the ones that tried that one, was not really solving. So things are improving, but still a lot that we can gain. And we see people, CAR-T companies going into to Japan, China, or US, but not going into Europe because it's complex and long.
So net, the sort of ICH kind of harmonization that's happened for small molecules and large molecules globally hasn't really caught up in cell and gene yet in Europe. Was that the case?
Not really. I mean, another piece of news that will come hopefully in 3 weeks from now would be a guideline, a European guideline by the CAT on investigational ATMPs. So hopefully in that one we will have some minimum requirements there for exploratory and confirmatory trials. So hopefully will help reviewers in the member states aligning in the review. And also there's some news on what the EMA is calling ATMP curriculum. So it's a full training program that will be rolled over from the EMA to the member states, to assessors and inspectors. And again, that is another nice initiative to educate and consolidate the feedback and ultimately accelerate the approvals, hopefully.
Just one question, which wasn't on our original list, but came up in the previous session about the long-term. Forget who raised it. About the long-term follow-up required in the US. Is there very similar requirements in Europe?
So again, in Europe, it's this idea on the risk-benefit approach, and it's really that you have to make your own case. But it's true as well that we have some benchmarks in there of 10 and 15 years follow-ups, and it's pretty much the same expectation. Although you can build your own case in a way as well.
One thing that might be interesting to us as an industry, is to begin to explore whether there might be opportunities to provide some kind of an overall platform system for long-term follow-up on cell and gene therapies, because many of the companies and indeed investigators in AMCs that are getting to the point of being very close to therapeutics, I mean, the load on any one individual institution to think through the regulatory, the legal, the technical of setting up a long-term follow-up is quite challenging. So maybe as an industry, as part of ARM or the foundation, we might begin to think about what some kind of whether it's possible to envisage a sort of a universal, it's a very grand term, but a universal data platform that is available to all. It would have to be, largely standardized, but configurable for each particular therapy. But maybe we'll pick that up as a talking point at the end. Have there been any discussions about that from any of the panelists? Have you been aware of any discussions like that?
So I'm aware, ARM is organizing another nice initiative on, registries, US and Europe. So that would be, a nice platform to canalize those level of discussions. That definitely is a topic that ARM is pursuing. Moving
to Brainstorm, Joe, you guys did phase 1 and phase 2 in Israel, and then phase 3 in the US. Why? How did it work out? What were the lessons learned?
Yeah, so that's correct. So our initial proof of concept clinical studies were done in the first 26 patients at the Hadassah Medical Center in Israel. And then we did a phase 2 study at 3 centers here in the US, the Mayo Clinic, Mass General, and the University of Massachusetts Medical School, in 48 patients. And then we essentially took that experience and that was using a single administration of our proprietary technology platform called Neuron, which basically utilizes an innovative autologous cellular therapeutic approach for isolated mesenchymal stem cells, which are induced to secrete neurotrophic factors. So we essentially use those learnings, CMC capabilities, and now we have a phase 3 pivotal study enrolling at 6 centers in the US. So I think what I've been impressed, we're going to touch upon this later, but I joined Brainstorm this past summer after 4+ years at Juno. So I have the unique experience of seeing Juno in the early days when it was an academic process that necessarily had a lot of pitfalls in going mainstream. It was an academic cellular therapy process. Could we scale this? Could it work? I have to say I've been particularly struck by the company's ability and excellence really in CMC readiness. It was touched upon in panel 1. I sort of see it as sort of our secret weapon. I mean, we've been working on mesenchymal stem cells for greater than 10 years in Israel, and these are highly trained R&D scientists from Tel Aviv and the Weizmann Institute. So these are folks that come out of world-class cellular therapy institutions like Zela Geshar's lab, some of the early pioneer work for CAR-Ts. So essentially that experience we used to now enroll in a phase 3 study now, which we recently announced in August. We just cleared an interim pre-specified DSMB threshold in the first 81 patients. And the study was basically deemed to go on without interruption, so no serious adverse events. So it's really all systems go with respect to preparing for BLA and US FDA approval moving forward.
Just working on this idea of there may be geographic arbitrage. Clearly we'll get to Hardy and Japan and the regulatory framework in a second, but is there any merit in other companies doing what you're doing in doing early work in Israel and then coming back into the States?
Well, it's an excellent question, and I think as we sort of evolve in this discussion, I think we're probably going to talk a little bit about some of the work we've done in terms of that CMC readiness and ensuring efficient tech transfer. Quite frankly, in the early days of Juno, we faced a lot of issues with respect to tech transfer and taking this academic process and translating it into commercially scalable product production. And we've really taken an approach at Brainstorm to have a hands-on approach. We've basically employed our Israeli-trained scientists at these centers of excellence. So we executed a contract with the City of Hope in March of 2017 to manufacture Neuron. And then more recently in July of 2018, we executed a contract and we'll be working with Dana-Farber. So we have the benefit of these centers being actively involved in the early days and sort of pioneering that CAR-T work. Steve Foreman's lab at City of Hope, and then a lot of the great work at Juno and other places were done in Boston at Dana-Farber and Mass General and other. So a lot of that infrastructure and sort of the clean room, CMC suites, things that are in place are modular enough that we could actually hit the ground running quite efficiently in manufacturing mesenchymal stem cells. I mean, really for us, a lot of the only hurdle in scale has been the ability to manufacture and scale quickly. So that's why we made the decision to add on these academic centers initially as we get ready for a commercializable process.
A little bit of a loaded question, but how did you choose City of Hope?
I'm new to the company, so I don't have all the backstory there, but I would think from a purely intuition standpoint, I mean, it would be what I just alluded to. Steve Foreman and a lot of the folks at City of Hope had pioneered some of that work early on, and they clearly know. I mean, Mike Jensen, who I worked very closely with at Juno at Seattle Children's Research Institute, he also, sort of set the standard with how cellular therapies were going to be done at academic centers, and then, had the foresight to think about scaling toward FDA approval. So it's that, I remember early in 2014, I attended every presentation. The presentations were more about engineering cellular therapies, and it was all, big terminology. It wasn't necessarily mainstream, and it really sort of evolved over time. So I think it's partnering with those institutions that had the foresight to think futuristically.
I know, because we speak to a lot of STEM and cell gene therapy companies, that there's a big queue of people waiting behind you to get to City of Hope.
Correct.
So one last question.
We're definitely fortunate to be there.
Yeah. One last
We have a question from the audience.
Oh, good.
So I'm with Brainstorm Cell Therapeutics as well. So my name is Mary Kay Turner, and I do our government affairs and advocacy work. And just to add to what Joe said, one of the reasons, and really important why we chose City of Hope, was because the California Institute of Regenerative Medicine is our partner and funded our phase 3 trial by the means of $17 million. And so it's that connection with the Alpha Clinics and with City of Hope.
Very good.
Thank you for that clarification, Mary Kay. Thanks for saving me on that. Absolutely a fact.
And that was the answer I was looking for. Okay, so one last question, because I think from to have all the details, but I understand the phase 3 recruitment's going well. Do you have any particular? I mean, there've been considerations in the previous panel about barriers to recruitment, and most trials don't make enrollment on time. I mean, what's been your experience of the value proposition to patients and the uptake?
Yeah, I think I alluded to it earlier. With respect to barriers for recruitment, I mean, I think we have the fortune of being in phase 3 pivotal development with a cellular therapy, so in a huge unmet need of ALS. So really, patients do the homework and the research and are basically going to their physicians to talk about this trial. So I think I meant what I said earlier, in that the scalability and the CMC infrastructure were really the things that we had to focus on, because I think patients believe in the clinical promise of this potential therapy.
So, if there's some kind of fictitious world in the future where everyone goes to Israel because they get all the CMC really sorted out early, and then they go to City of Hope because they've got the state-of-the-art manufacturing. From a regulatory perspective, Japan really stands out. So Hardy, with the Sakigake approach to development and early commercialization, can you tell us a bit about that and how you've leveraged it?
Yes, so Sakigake designation is practically like a fast-track designation. Once you have the stamp on it, you can go to monthly consultation to Ministry of Health, especially cellular therapy company needs to consolidate the manufacturing side of it as soon as possible while you're running phase 2 or phase 3 trials. So it's really helpful. And also, they're going to shorten the time frame for approval from review period from 1 year down to 6 months. So it's really accelerate the approval process. And in order to get the designation, you should have some proof of concept data, and also the therapy should meet critical unmet medical need. And we have got the designation for multi-stem technology license from Athasys for stroke. And we are running phase 3 trial right now, and then 1 more year to be fully enrolled, and then an additional 1 year to get it approved. So we are hoping to get this pivotal study and all the product approved in 2 years' time, which is really helpful. And I'd like to mention one more thing about the regulatory framework. Sakigake is good, but the real strength is the clinical research system in Japan. Clinical research is different from clinical trials. And we started world first iPS clinical research back in 4 years ago with the autologous approach, patients doing well. And we shifted towards allogenic approach, HLA match 1, and 5 patients enrolled, and there was some side effect due to the change of the formulation issue, and we fixed the problem. And later this year to beginning of next first quarter, we are suddenly starting the pivotal study for iPS AMD study. And we are hoping this product will be the world first iPS product to be approved. But the key message here is, by combining academic-based clinical research, which is not categorized as phase 1 or 2, but we get enough information, and we can design sufficient pivotal study, and then you can suddenly go into a pivotal study. That's the unique beauty of the Japanese system. Then the question is, can we use the data to accelerate the approval in the United States? That's a big question. But the answer is I don't know yet. But this is my second biotech company. With my first company, we run academic-based clinical research for small molecule for 60 patients, and that data was good enough for us to skip all the phase 1-2 trial for US trial. So then we could, back then, we could start a phase 3 trial in India and the United States for pivotal study. So we don't know. We might be able to do the same thing.
So, but there's at least one precedent for using the Japanese framework to accelerate development in other regions.
That's right.
Okay.
And also, sorry, one more comment. Athasys has publicly disclosed, but by collaborating companies like us, FDA has given the commitment that they'll be able to use our phase 3 data from Japan as one arm of the study for US approval. So Athasys could reduce half of the cost for running 2 pivotal studies in the United States. So that's something you want to keep in mind, I guess.
Has anyone in the audience even had a tentative discussion about the potential to use the Japanese framework as a first step in the early stages of development? Is that something that comes up these days? I think that we're very intrigued in trying to get some kind of a policy paper or some kind of an evaluation. I know that, Katie, you were telling me that Commissioner Gottlieb's remarks last Friday, and their remarks, and we've got to wait and see what policy and regulation is, but they were very positive about trying to include, particularly in the cell and gene space, evidence from other regions. So things may be moving in that direction, and there may again be some kind of an arbitrage to leverage. And another thing I wanted to ask you is, you really focus on partnerships. It's a big part of Helios. Can you speak to that a little bit?
The part, sorry?
Your partnership strategies.
Yes. So we are open for partnership. Our strength is, so basically we are about to run 3 pivotal studies in Japan. All of them, we are hoping to get all of these 3 pipelines to be approved in 3 years' time. And we will be building Salesforce, and we are open for collaboration in licensed technology from United States or elsewhere into Japan. We're gonna fund the trial. We're gonna pay the whole trial, and then give the data back to our partner. I think it's a really creative and unique way. Especially in Athasys' case, we became the largest shareholder of the company too, and I'm serving as a board member at Athasys. So there are a number of the things we can do to help each other. And we all want to accelerate the program. We all want to reduce the cost for clinical trials. And this is actually really is a global trial. And then if we can help each other, I think that's great.
One thing that came up on the previous panel, and I was curious just to get the European and Asia perspective, I mean, it's not meant to be a pejorative term, but as a shorthand, the discussion was around like garage iPSCs or garage CAR-Ts, where maybe organizations that are less regulated are beginning to offer services which seem to have better access and less procedure than clinical trials. And again, at a sort of a much more formalized level, the potential that academic medical centers could turn into small therapeutic companies. UCSD, maybe that wasn't what was being suggested, but certainly others are in discussions about, why don't we keep the vector? Why don't we get a Miltenyi box? Why don't we do vein to vein in the clinic? Get through with some tens of patients to the point where it's close to being a therapy, and then do treatment locally. So is that something that you're seeing in Europe and Asia too, or is that a particularly US approach?
If I may speak via Europe, we don't have that many. Still some activities. We don't have huge, I think, unregulated and at least in some member states, this has been, targeted and followed closely by ministries of health, and we've seen some of them being closed. Having said that, we have some parallel systems, and I wouldn't say unregulated, but it's something apart from the normal development and centralized procedures, things like specials, hospital exemptions, things that you might have heard in the past that also are in a way, competition and causes also some problems, especially in orphan or niche indications as well. So that's another one that we try to follow, because can jeopardize also development and recruitment in clinical trials as well.
In Japan, it was unregulated. Under physician's law, doctor's law, they could do anything. But now it's regulated, so some of the clinics are basically shut down for those new activity, new law in place.
Robert?
Yeah, I was going to comment that I don't think there's a compromise on integrity or data collection. I think the accelerators are driven by time and the review process. So I think being on the other side of the story with Hardy vis-a-vis the Athersys product, I found that all of the feedback we had from the Japanese regulators were on par and equally tough as we had from Europe or the US, North America. So I think there's tremendous opportunity in this space to look at accelerated regulatory approval as the mainstream platform for development within a company. I think to go to the point that Dan raised about universities building GMP centers to facilitate translation, what you're starting to see is a drop in the small biotech as a player in value creation. You're seeing the universities are building the story big enough to drop out to mid-size, either to healthcare or mid-size biotech, and translation is accelerating there. So I think the new regulatory environment we're working in is having incredible impact on our industry. I think Hardy also underplays his role in this. I think he saw this roll out about 5 years ago and positioned himself to take advantage of this and to enable it across the community. And I just will applaud you for having that vision.
Moving into manufacturing, so that that's a massive insight, right? The whole, the headline, if one was to be an irresponsible journalist, might be biopharma disintermediated from cell and gene therapy production and provision to patients. It's not going to go that far, but there's something big happening. And as well, just to note, it is the well-founded, well-established AMCs with top-tier research that don't necessarily need to put it out to manufacturing plants in Puerto Rico anymore. It's also foundations like the Parker Foundation that are looking at various indications and hoping to build their own end-to-end system at scale, including large-scale manufacturing plants here in California. So there's something big and interesting happening there we've all got to keep an eye on. Robert, if I could stick with you, in the last panel, the discussion of autologous and allogeneic was touched upon again, but you've actually have a perspective from a technological perspective about how to engineer out the immune response in allo. Could you speak to that?
Yeah, I'd be happy to. So BlueRock is developing cell therapeutics for the heart and the brain, and our platform is driven by a pluripotent stem cell biology and gene editing capability. And so one of the fundamentals of our approach is that our cells can be practiced or enabled for allogeneic use. And so we've gone after that by understanding that new knowledge driven from the immuno-oncology field plus new technology capability from gene editing allows us to edit the receptors that are responsible for immune sensitization and cell rejection. So we have a relationship with a company, Universal Cells, where we're in the process and have used the beta-2 microglobulin locus as a target for manipulation of immune receptor genes. So we use that locus to knock in a safety switch for our cells. We use that locus in the same cell to knock in a polymorphic HLA gene that can block NK celling and an innate immune cell rejection. So the outcome of that, by ablating beta-2 microglobulin, is we keep class 1 HLA from the cell surface. So we're preventing T cells from recognizing the product. We all know that in the absence of HLA engagement, NK cells can recognize a cell as foreign and reject it through NK-mediated killing, and that's why we're using the polymorphic HLA-E molecule. So we have very high hopes about that. It's borne true both in our in vitro and our in vivo experiments that we've performed to date. And so we're quite comfortable with the gene editing approach managing acute rejection responses. I think where we have more emphasis in terms of our research program and bringing this to proof of concept is around chronic rejection and understanding that this mode and this approach can block chronic rejection. But in addition, we're also very interested in immune regulatory cells. And I think, to go back a little bit to Dan's talk earlier, it's clear people are developing dendritic cells from iPSC as well as regulatory immune populations from iPSC. The CAR T programs that we've all seen function so well in the adult community are moving to a pluripotent base for cell manufacturing. So I think we also see immune regulatory cells as being able to contribute to our ability to have our products engraft long term. So long story short, in addition to gene editing approaches, we're also very, very interested in immune regulatory cells as a new paradigm, edited immune regulatory cells as a new paradigm for tolerance induction.
Blake, if you've got a perspective on, again, from this very early stage cell engineering point of view, how to start using iPSCs, particularly neurodegenerative related, in early toxicology to try and cut out some of the later, maybe reduce some of the risks in later stage development.
Yeah, correct. So, sorry. We're really approaching this from the iPSCs as a tool to look at early risk mitigation. So again, this is an example where the iPSC-derived tissue cells, such as neurons, microglia, endothelial cells, cardiomyocytes, can really be used as a relevant cell type to understand potential toxic interactions with the therapy. This really stems from some of the events that happened in the MAGE A3 trial done by Adaptimmune and Immunocore, where there was off-target toxicity that had a cross-reactivity with cardiac titin. This toxic effect, and I believe it was the first 2 patients, ended up dying in the clinical trial. And this cross-reactivity was not picked up by any of the other preclinical models that they used, whether they were differentiated cell lines, de-differentiated cells from humans, or non-human species. Right? It was that functionality of the cell type really functioning as a normal human cell would function that was essential. And so this we're seeing as a way of really doing prospective risk mitigation, as well as potentially looking for batch validation or batch qualification of these therapeutics as they come off. If there's any variability in there, again, these can very easily be run across iPSC-derived tissue cells.
One theme that's been coming up more and more in these panels and in our work too is, again, to put it very simply, this isn't really personalized medicine, it's more personified medicine. And I love one of the terms that was used in the earlier session. It was living drugs. These are living drugs. And so the condition of the innate immune system upon harvesting, say, T cells, the details of the production process, the immune state of the patient when they receive the therapy, and then the clinical and economic outcomes of using the drug, they're all viscerally tied back to that origin, that person, that point of manufacture, at least for autologous. And even in allogeneic, it'll still be back to that very specific part of the process. Do we have systems and processes that properly track the provenance and the chain of custody of cells from origin to patient from vein to vein? There might be an answer in itself.
I think it depends upon the complexity of the study, and just based on my personal experience, I've worked in environments where we've had a product that was made and carried in a centralized distribution center. At time of patient need, we would reformulate so that the dose was set, viability was set, and we would drop ship to the enterologist office. And at that site, there was no intervention by staff other than hanging the bag and IV infusing the product. So we obviously built that out so that we could control quality of the product going in, and we were moving outside of stem cell centers at university hospitals and moving to a physician's office. So I think there's an awful lot that still needs to be built out on in terms of how we can deliver the product with confidence that it's dosed and potent to go into the patient without having to have an individual on site. I don't think that that's been clearly, I think we've had limited experience community-wise in that space, and I don't think we have good solutions today. But I will pass it over because I know you can comment.
Yeah. So I think at Brainstorm, I'll comment on this. I mean, I think we've worked very hard to work on the supply chain and identity aspects of the entire process. And we're to the point now where we end up with a 4 ml, basically syringe-ready product that could be administered on site. So through our end-to-end, our process to create the MSC NTFs is about a 7-day process to create the commercial ready, and it comes shipped 2 to 8 degrees. And we've partnered with a company called QuickStat for the supply chain logistics to accomplish that.
This is interesting. I mean, this could either end up, well, it'll clearly start off by being a competitive advantage for certain companies because you have that, vein-to-vein provenance. In the end, one might think that as an industry, we really have to work with technology companies and with regulators to make that a requirement, because the wrong cells going to the wrong people just doesn't work out well.
Yeah, I mean, I think on the CAR T side at Juno, I mean, we had all kinds of grand plans with respect to apps and all the supply chain identity. I think now, since Kite and Novartis have been approved, and as Juno's will be third to market, I think a lot of that supply chain logistics has been worked out. So we'll certainly look to rely upon some of those aspects and learnings and apply them appropriately.
So we seem to be envisaging a world where, not only when we're going for registration in different markets, but even during development, therapies may be moving around the globe for different parts of the development. Gisèle, you see everything everywhere in helping many companies in tech transfer. Couple of questions. Question number one is, what are some of the very common themes and stumbling blocks you see many companies falling over? And then is there a more systemic approach that could be a guideline to be adopted, which will help various early stage companies plan better for that tech transfer side of things?
Yeah, so at MaSTherCell we are completely dedicated to development and manufacturing of cell and gene, ex vivo gene therapies. So for the last almost 7 years now, we've gone through probably more than 20 tech transfer, and only 40% of them intra-European. So I don't know if we can call that non-international tech transfer, because as Maria explained, we have also major differences between our regulatory authorities between the different countries in Europe. So I would say the international tech transfer it might only be. It is not a major challenge. It is just another layer of attention you have to put into details, like, of course, communication and anticipation. So in terms of communication, we cannot avoid putting more attention in language and time zone differences, and also, of course, cultural differences. So it is worth spending a bit of time ahead of doing the actual operational tech transfer to work together and to make sure that the communication is open, transparent, that you manage to work as a single team, And one good way to do it is to work together through risk analysis, risk assessment, gap analysis, because there are definitely gaps, between regulations from different countries, definitely the type of raw material or ancillary materials, as you call them here, that you put into your process. I mean, some might be compliant here and not compliant in Europe, and this is something we definitely need to identify before starting, because they might need to be replaced, which then calls for additional planning for testing and for maybe comparability studies, where you were just hoping to go through an easy tech transfer from phase 1 to phase 2 or phase 2 to phase 3. You might find yourself obliged to run additional testing and validations. But it's nothing that cannot be done. It's just that it's better to anticipate and to do that together with the customer, because it's a very important timing and reflection that you can do together. And then yes, as I said, you need careful risk assessments about all the gaps you could have between the different countries and regulations.
Thank you. In the last 5 minutes, I just wanted to touch on a couple of innovation topics, and one is, development's development. It's different for cell and gene therapies for many of the reasons we've discussed. But the other thing that's happening is more and more use of real-world data. The regulators are becoming more comfortable with it. The provider systems and the patients are becoming more competent at managing it and providing it at the right kind of standard to be relevant to clinical research. Robert, I know that one of the things that you've been looking at is using natural history studies, looking at biomarkers, and being able to anticipate accelerated approval, accelerated regulatory pathways through being able to devise surrogate endpoints. So is that something particular to BlueRock, or something you think is going to become more pervasive across the field?
I think it's going to become much more pervasive. And so as a backdrop to your comments, BlueRock is developing cell therapeutics for treatment of Parkinson's disease. And in our overall structure, our lead PI had begun a natural history study. So patients that will enroll in our treatment study will have had an extensive, greater than 1 year, read on disease penetrance and their decay with standard of care. So it's clear that that's advantageous to us so that we can measure patient response against our own individual baseline. But I think what's more important underneath that is realizing that machine learning is opening the door for us to interrogate data much more aggressively than previously thought. And one of the hallmarks of the RMAT, the Accelerated Regulatory Approval Process in the US, is the use of surrogate endpoints for efficacy that you then use in a registrational study. So moving into RMAT as a cornerstone or a standard process means that you have to amp up your CMC capability, because you're going into a phase 1 study that's going to generate data that would lead you to a registrational study, and you have to know that you can solve your commercial manufacturing problems. The other thing that it does, and this goes to the value of the natural history study, is that it lets you begin to collect biomarkers based on blood sampling and genomics technology that at the outset you may not understand the correlation and linkage, but you can build a database and with machine learning get some tight and surprising correlations to use in your registrational study. So we've incorporated machine learning across the board in analysis of our bioprocess, single-cell RNA-seq analysis to tell us where the inflection points in our process are and how to use that information to get the cells we want or eliminate the cells we don't want. We've used machine learning to build potency assay strategies where we can compare a composition of a product and understand how it performs in preclinical studies, and by looking at failed and successful products, hone in on the markers that really represent true surrogates for potency. And then finally, we really see that clinical stratification of patients post-treatment with these more sophisticated analysis tools can give us a much higher likelihood of success going forward. And in that process, if we can stratify the population of patients that responds to treatment, it means that we can go forward in registrational studies that are much tighter, less cumbersome to execute, and don't represent the huge financial hurdle that many of us are interested in. So if you look at what we're building in terms of products for the heart and brain, you're talking heart failure and Alzheimer's as a target. Those have traditionally had 3,000 or greater patients enrolled in pivotal studies. You know, we're now looking at a world where we can cut that by 10 and understand that learning much, much earlier. So I'm obviously an incredible proponent of bringing machine learning into our development process.
Blake?
Quick question. In the last 10 seconds, quick question, actually for the panel here, in getting new innovation in front of and accepted by the regulatory bodies. So we've been involved a lot with small molecule and getting the stem cell platform in front of the regulators and running that up through, you know, the ICH harmonization process and whatnot. And it literally takes years. And now we're seeing customers being asked by the regulators to provide some data, safety data, using the stem cell platform, but how to actually move that to some rapidly forward so it actually has an impact in a short amount of time, rather than 5 or 10 years down the road. Are there any thoughts on that?
I'm afraid I'm going to have to. You know, on the Muppets, when they got pulled off with the hook, that's what's happening to me now. But thank you very much to the panel. And then, but before we applaud them, I want to just give you some final thinking. We seem to have had 3 or 4 areas of arbitrage: regulatory arbitrage with Japan, CMC arbitrage with Israel, a unique disintermediation that's happening with academic medical centers, an opportunity for pre-competitive toxicology, an opportunity to have a platform for long-term follow-up that the industry shares, and also a more unified approach to getting surrogate endpoints from natural history accepted. There's a lot changing. Thank the panel. Thank you very much for attending.